Hello and welcome to the second lecture, or session 2, for chapter 4 on tissues. Last time we were together, we were talking about the general characteristics of tissues, and we delved into epithelial tissue, looked at the structure of epithelial tissue, we talked quite a bit about the basement membrane. Today, we're going to continue that conversation. We're going to talk a little bit about the characteristics of epithelial tissue, the different types of epithelial tissues, its functions, and then we'll do a survey of all of the different kinds of epithelial tissues that you find in the human body. So some of these you've already been introduced to in the lab, but there will be some new ones. And we're going to really not spend a whole lot of time on describing and looking at each of these different types. We're going to get the basics, where they are, sort of their basic structure and their function, and then move on to the next one. So let's begin with the characteristics of epithelial tissue. So characteristics. Characteristics. And we've already made this list in the lab, so this is a bit of a repeat, but let's go ahead and do that anyway. So epithelial tissue is found in continuous sheets. And at least this is for covering and lining epithelium. And these sheets can be a single layer or multiple layers. Another characteristic, and this is true for both covering and lining epithelium, is that we are looking at densely packed cells. So densely packed cells. That means that there is very little extra cellular material. Okay. Number 3, they're going to be held together. That is, the cells are held together by special cell junctions, and these are things that we've talked about already in the first lecture, session 1. Okay. Number 4, this tissue is avascular. So it does not mean that there is no blood supply nearby. There is. We can find it sometimes in the basement membrane and sometimes just below the basement membrane in the connective tissue underlying the basement membrane, but not within the epithelial tissue itself, not within the cells that make up epithelial tissue. Okay. Number 5, supported by a basement membrane. And we've talked about that quite extensively in the last lecture. Okay. Number 6, highly mitotic. And that means that we have a high rate of cell division in this tissue. And this is often the case, especially in tissues that are under a lot of wear and tear and under a lot of stress, such as their skin. Okay, so highly mitotic. Number 7, it's going to combine with nervous tissue to form the special senses. And to give you a sense of what we mean when we say combine with nervous tissue to form special senses, let me just give you a list of things that fit that description. So, for example, we have the sense of smell. And here we have the olfactory membrane. And you're going to run into this in 40B. This is in the roof of the nasal cavity. And it's a combination of epithelial tissue and specialized neurons that allow you to smell various scents. Number 2, another example would be hearing. So again, you're going to run into this in 40B, and we'll talk, or you will talk, I will not, talk about the structure of the ear and the inner ear, where you're converting physical movement or air pressure changes into an electrical impulse that you interpret as sound, and this is happening as a combination of nerves and epithelial tissue. Number 3 would be vision. Once again, you're going to run into this in 40B. So vision in terms of the retina and the eye, that's a combination of epithelial tissue and neurons. Okay. We also can talk about taste buds. And taste buds are located on the tongue, combination again of epithelial tissue and nervous tissue. And just to finish off our list, we can look at things like touch. Once again, here we're talking about, of course, the skin, which is epithelial tissue and nerves. Okay, so that's the characteristic of epithelial tissue. As we've seen before, there are 2 types. Number 1, covering and lining epithelium. And number 2, glandular epithelium. So for most of the following lecture, we're going to be covering the first category, that is covering and lining epithelium. And towards the end of the lecture, we'll talk a bit about glandular epithelium. So we'll cover both kinds in this lecture, hopefully. Again, I'm not sure how long the material is when I actually lecture in this format. So I might break it off before we actually get to this part, but we'll see. Let's talk a little bit about the functions of this tissue. Number 1, protection. And a classic example of that is the skin, although it is not the only example. So when I give you these examples, they're typically not exhaustive lists, but enough to give you a sense of the idea of what that particular statement or principle is talking about. So number 2, secretion. And here there's a bunch of examples we can talk about. Digestive enzymes. Of course, this would be in the digestive tract, but also mucus, another example. Hormones, okay, that would be glandular epithelium, and a bunch of other possibilities. So secretion. Number 3, absorption. And we can talk about here nutrients from your meal, ions, minerals, glucose, all of that. And that, of course, it would be taking place in the digestive tract. That's the main area where we see absorption being a function of epithelial tissue. And the final example, or the final function, I should say, is going to be excretion. So it excretes. And a very good example of that is the urinary tract. And that you actually tackle in 40C. Okay. So those are the general functions. Now, let's start with covering and lining epithelium. And let's talk about the classification. How do we name epithelial tissue? And you're already, hopefully, very familiar with this system. As you know, it's going to be based on two characteristics. And these characteristics are cell shape, and number two, cell arrangement. Let's begin with the last one first. That would be cell arrangement. And here we can talk about a number of different systems. So we can talk about a single layer, one layer, and that would be identified as simple. One layer of cells, and the kinds of functions that are carried out by a single layer of cells would be diffusion, osmosis, filtration, secretion, and absorption. One single layer of cells. Another system would be many layers or multiple layers, so more than one layer. I think my pen is about to run out of ink. Fortunately, I have a standby. Okay. More than one layer, in which case we're going to call that stratified. And so this is all about protection. Although there is some secretion involved in stratified layers of epithelial tissue. So most of it is about protection. Okay. A third system, which is really not a separate system, it is in fact a single layer of cells, but it doesn't appear to be a single layer. And so we identify that as pseudostratified epithelium. And I just think that's a very unfortunate turn of events, because it just simply is confusing. If we just stuck with the two systems, this would be under simple, but that's not what we are given. So let's talk about pseudostratified. Epithelium. And pseudostratified epithelium, and this would be number 3, of course, is actually a single layer. It's actually a single layer, but appears to be more than one layer. Okay. And that is because not all cells reach the apical surface. So if you were doing a little sketch, you would have, this is my basement membrane here, you would have cells that are actually short of and don't reach the apical layer. And you would see something like this. And when you're looking through the microscope, you might mistake this for stratified, because that little technique that I talked about in talking about counting the number of nuclei, this would appear to be multiple layers of nuclei, and that would only be because this cell, which is shorter than this cell, its nuclei is further down, this one is a little bit higher up, and it creates this stacked appearance. And in fact, it's just a small cell that doesn't reach the apical surface. And this type of tissue is secretion. And we're talking about mucus, which also means that we're talking about protection. Okay. So that's the arrangement part of the classification of epithelial tissue. There is the second part, which is cell shape, which we'll address right now. So cell shape is also going to play a role in naming epithelial tissue. So cell shape. And there are a number of different shapes. So we can talk about squamous. And these are flat cells. And flat cells are good for rapid transmission of material. Two S's. Someplace like the lungs, for example, in the respiratory membrane. Number two would be cuboidal. These are cube-shaped cells. And here we're talking about secretion and absorption. Okay. Number three, columnar, which is column, tall, and cylindrical. That's probably a better way of talking about. Okay. And this is about protection. And secretion. And some of this tissue is ciliated. Not all columnar epithelial tissue is ciliated, but some is. Okay. And the final and last category is transitional. And this is a type of tissue or type of cell that transitions between cuboidal and squamous. And this is all about increasing surface area. Alrighty. So now we're going to start our survey and go through the different kinds of epithelial tissue. I just jumped the gun a little bit because I did want to spend a little time reflecting on what we just wrote down using this overhead. So here's the two arrangements. We have simple and we have stratified. One layer of nuclei, multiple layers of nuclei. So I think I've mentioned this before. One thing to keep in mind as you're looking at this tissue and trying to figure out whether you got simple or stratified is that stratified is very seldom just two layers of cells. So if you're going to have multiple layers, it's going to be more than 2, 3, 4, 5, 6, and sometimes many, many more than that, depending on the type of tissue you're looking at. So it's not going to be that confusing. You'll either have a very simple layer of cells or many multiple layers of cells when you're looking at the arrangement of cells. And then, of course, we have flat cells, squamous, cuboidal, safe shells, and columnar. So those are our characteristics. Notice that what's not being shown in this image is the pseudostratified, which is unfortunate, and transitional. But we've seen a lot of that in the lab, and you should be now also, of course, testing yourself on that material in the practice exam that I've already posted in Canvas. All right, well, let's take a look at, as I said, we're going to do a survey of the various types of epithelial tissue, and I would urge you to look at this table. This is where I'm getting my information. Table 4.1, pages 112 to 118. So there's a very nice table in the textbook which lists everything that we are about to talk about, and all the information, if you somehow miss it in my lecture, is there. So let's begin with the first tissue, which is simple squamous epithelium. And we'll talk about location. So a variety of places, blood vessels, lining the heart ventricles, serous membranes. Once again, please note that I'm not giving you an exhaustive list here. I'm giving you some examples in a variety of different places. Now, one of the things that often comes up with students is terminology here, because you will hear or see a term like this, endothelium, and you'll look at that and you'll say to yourself, well, that sounds an awful lot like epithelium, but it isn't actually epithelium. It's something else. It's called endothelium. So what am I really talking about? Well, this is in fact epithelium, and it's often associated with simple squamous epithelium. So this kind of word usage that I'm about to talk about is most often connected to simple squamous epithelium, which is why I'm talking about it here. So endothelium is found within blood vessels. So this is within vessels. And the best example of that is lining a blood vessel. And it is generally identified as such. So you very seldom actually see the term simple squamous epithelium applied to identifying cells in lining a blood vessel. So if you had a blood vessel, and blood vessels generally have some thickness to their walls, okay, and you would normally see a series of flat cells that are lining this, each with their own nucleus. You get something like this. this kind of arrangement like that. And that would be identified as endothelium. So inside a blood vessel. All right. The other term you'll often see is mesothelium. And here, primarily what you'll see that associated with this is the serous membranes. So this will be lining body cavities, such as the abdominal pelvic cavity. And that terminology, mesothelium, is often referred to as when you look at those cells. Okay, so that's location. Let's talk a little bit about function. Function is filtration. And here we can look to the kidneys as an example. Diffusion. The lungs. Perfect example. Secretion. And this would be serous membrane. You're not going to find this type of tissue, so it's not found in body regions subject to stress. And here we're talking about mechanical stress, physical forces acting on the surface. Okay? Not found there. It's too fragile. It cannot hold up to any kind of real physical stress. So these are pretty well protected spaces where we find the simple squamous epithelium. Okay. Let's take a look at an example. So here is, and they're using as our example in this case the serous membrane. In fact, the peritoneum, which is a serous membrane, and this is the mesothelial lining of the peritoneum. There's that word denoting the cavity, or lining the body cavity. So all this stuff that you see in blue, the top surface of that would be the simple squamous epithelium. Okay. All right. Let's talk about simple cuboidal epithelium. And there's a U in there. Okay. Simple cuboidal location. Number of, we're going to see the ovary, find places within the eyes, that is regions of the eye, not the entire eye. The kidney tubules, smaller ducts, and some glands. Function here is secretion and absorption. Simple cuboidal epithelium. Let's go ahead and look at an example of that. Bada bing, bada boom. There it is. And here we're using the pancreas as our example. So You'll learn about this in 40C. We'll do the digestive tract 40C. Pancreas is part of that system. It produces a lot of digestive enzymes. And there are some major ducts that run through the length of the pancreas, and those ducts are lined with simple cuboidal epithelium, as you can see in this image. So basic structure, we've seen this in the lab. We don't have to spend a lot of time here. So that's simple cuboidal epithelium. Let's take a look at non-ciliated simple columnar epithelium. Let's talk location. Gastrointestinal tract. Some ducts of some glands in the gallbladder. Yeah. Functions: secretion, and absorption. Now, one of the great advantages of simple columnar epithelium, or columnar epithelial cells, I should say, as compared to simple cuboidal epithelial cells, is that you have much more room for the manufacturing of substances. So the size allows for higher levels of secretion than cuboidal cells. And this would be, you're going to see this in the digestive tract. So the digestive tract, as we've seen in our list, the gastrointestinal tract, is lined with simple columnar epithelium. Why not simple cuboidal epithelium? Well, we have two very good reasons for that to be the case. One of them I've just listed here. The larger cell allows for more production within the cell of like digestive enzymes that are going to be secreted into the digestive tract. The other great advantage of having a tall cylindrical cell as opposed to smaller cuboidal shaped cells is that you have a greater opportunity to create those watertight junctions, those tight junctions between cells. So if we take a look at, let's do a simple drawing to illustrate this. If I have a series of cuboidal shaped cells here, I have a relatively small surface area to form that watertight seal. On the other hand, if I put a columnar cell there, I not only get the advantage of more production space, but I have this longer common border between cells in which I can form that tight fit between them and therefore control the movement of substance across that epithelial layer much more efficiently than if I had a cuboidal shaped cell. Right? All right, let's take a look at an example then of this. And lo and behold, it is going to be from the digestive tract. Oops, grabbing the wrong pile. Hang on, I'll get it, don't worry. Okay. So, and we'll put this in English. So, small intestine, non-ciliated simple columnar epithelium cells. You can see the goblet cells. I've talked about those before. And I have a nice section in the tissues module that you should have downloaded and read. And we've been able to identify those structures too in the lab. So this is, and it seems like there might be cilia here, but remember, this surface has got those little, finger-like extensions, the microvilli, which creates that fuzzy edge, and that's not really cilia at all. All right. So, turn on some lights for us. Okay. All right. Now let's take a look at ciliated simple columnar epithelial tissue. And again, we're going to talk about location. So the respiratory tract, okay. Some sinuses. These are spaces within the skull that are lined with mucous membrane. The central canal of the spinal cord, and we talked about this in the first lecture, how the nervous system, that is the central nervous system, is produced from the folding of the ectoderm, creating a hollow tube. And the remnant of that hollow tube is called the central canal of the spinal cord, and it's lined with ciliated simple columnar epithelial tissues. The ventricles of the brain, which are simply an extension of that cavity into the brain. Okay. And the function here is movement of substances, not the cells, of something on the cell surface. So most of the time we're talking about mucus, the movement of mucus, but not always. We can also find that we can move the oocytes, which is the egg, in the uterine tube using ciliated cells, and cerebral spinal fluid. And that would be in the ventricles of the brain and the central canal of the spinal cord. Yeah. Alrighty, let's take a look at an example then of this type of tissue. And they are using the uterine tube. So lining the uterine tube is simple columnar epithelial cells with the cilia, and the beating of these cilia produces a current. So when the egg is released and from the ovary, it's caught up in the uterine tube, so it's brought into the uterine tube. A current is being created by the little finger-like extensions of the uterine tube here at the opening, but lining the uterine tube all along the line is this type of tissue right here, the ciliated simple columnar epithelial tissue. And those cilia are creating literally a current of fluid that draws the egg down and through this tube and into the uterus for implantation. So that's a perfect example of what we mean when we talk about ciliated and tissue being involved in the movement of substances. All right. Well, let's move on and let's look at stratified squamous epithelium. Okay. And here we have two forms. One is keratinized. And by that we mean that the cells contain a protein called keratin. And so this is within the cells. Okay. It is a protein. And it's designed for protection. It's a very hard protein, and it provides a lot of protection. We'll talk more about that when we do the integumentary system, the skin, and look at how the skin develops and the development of the outer layer of the skin. Okay, that's one type. And then, of course, the other type is the non-keratinized. Epithelium, and these are all this is always a mucous membrane, and this is a new term, mucous membrane. We haven't yet defined that term. We will towards the end of this lecture. We'll talk about what mucous membranes are, but mucous membranes obviously are going to produce mucus. There's a little bit more to that story, but for right now we can just look at it from that point of view. And this would be lining then the interior of the body, such as places like the tongue, the mouth, the throat, the esophagus, and places like that, non-keratinized. So some examples here would be mouth, the esophagus, the pharynx, also the vagina. By the way, the only place where we find keratinized is the skin. So that is the only place where that type of stratified squamous epithelium is found. Okay, so non-keratinized in a variety of different places. The function is protection. Protection, protection, protection. This is all what it's about here. Protection against things like abrasion. So what is abrasion? I bet most EMTs in the room can answer that question. An abrasion is a type of injury that is a scraping injury. So if you fall down and scrape your knee, that's an abrasion, as opposed to like a cut, which might happen if you were using a knife improperly. Okay, so abrasion. It protects them. It protects you from water loss. Also, ultraviolet radiation, which is a mutagen and can change the DNA of your skin cells. And it also is going to protect you from foreign invasion, from microbes. In fact, the skin is your #1 protection against disease that you have. Okay. So, stratified squamous epithelium. Let's take a look at an example of that. And lo and behold, they are using the vagina. So here's our stratified squamous epithelium. This is a mucous membrane. It will produce mucus. It doesn't look like it. It's not as straightforward as, say, simple columnar epithelium, where you can actually see the mucus producing glands, the goblet cells. But don't be fooled. This is a mucous membrane, and it does have mucus as part of its structure. So we see the stratification here. And, of course, this is protection. Nothing else is going on inside the vagina. We're not absorbing it. We're not secreting anything other than mucus. We can give the entire surface of this organ over to protection. Okay. So that's stratified squamous epithelium. Let's talk very briefly about stratified columnar. We don't see this very much. I don't want to spend a lot of time here. It's pretty rare in the body. and I never talk about it when I am teaching any of the other sections like 40B or 40C. So location, some parts of the urethra, and I do mention that when we talk about how the urethra is formed or produced, the structure of the urethra in 40C, but it's just a mention, nothing, I don't cover it in any kind of detail whatsoever. And some large excretory ducts, and also glands of the esophagus. Okay. Function, protection, and secretion. Protection and secretion. So let's take a quick look at that. And here we go. Stratified columnar. So they are using the esophagus, so the esophageal glands. And you can see there, it's not entirely clear, especially in this photograph here, that we're looking at stratified columnar epithelium, but that in fact is it. It's a little bit, perhaps a little bit better seen over on this side, but this is what we get. So this often happens with stratified tissue. It's only typically the upper layer of cells that form the shape over which it's named. So in this case, stratified columnar. So you can see that those are indeed column-like cells, but the cells underneath it that are forming underneath it don't appear to be that strongly column-like, only the top layer. But that was also true with stratified squamous. If you noticed, the stratified squamous, and we'll come back and look at that, in matter of fact, the stratified squamous, we also had a similar situation in which only the upper layer of the cells are actually flat, and the cells that are at the bottom region of this layer are not flat at all. So this is very common in stratified tissue, where we get this only the upper layer actually demonstrating the cell shape that the tissue is named after. And they name it because this is our functional layer up here. This is the layer that's in contact with the environment. These cells, these column-like cells, are in contact with whatever is in this area here. In this case would be the food, because this is your esophagus leading down to your stomach. Actually, let me take that back. These are glands within the esophagus, so they don't actually come in contact with your food, but they release the mucus, and this is the operating surface, this is the functional surface, and the cells that line that functional surface are the columnar cells, as you see right there. Okay. All right. Stratified cuboidal. Epithelium. Okay. Location. Ducts of adult sweat glands. So this is a developmental process that happens and only found in the adult sweat glands. Parts of the male urethra. And why just the male? It's because the male urethra is longer than the female urethra, because we have a penis, and that extends the length of the urethra. And then ducts of esophageal glands. So we have a mixture in the esophageal glands of both stratified columnar and stratified cuboidal. Function is protection and some limited secretion and absorption. And so let's take a look at an example here. All right, so again, they're using the esophagus, and this time we're the duct of the esophagus rather than the actual gland producing area. And so you can see stratification here. So this is the exception to the rule I mentioned earlier, where if you have stratified layers, they're typically more than one layer. So stratified cuboidal is the exception to that. And we can see just 2 layers of cells there. And it's sometimes difficult to actually know. It's clear here. There is a very clear picture of 2 layers of cells right there. Okay, so stratified cuboidal epithelium. And let's see, how are we doing? Checking how far we're going to go here. Yeah. I think we're going to go all the way up to, but not include glandular epithelium. We'll stop there because I hear the buzzer. My dinner is ready, and I'm definitely not going to give up dinner for you guys. So that's all right, though. You'll understand, I'm sure. So transitional epithelium. Right. Location, only one location. Urinary bladder. Now I'm about to make myself a liar here, but I'm going to explain why I'm going to give myself a pass on this. So let's just take the urethra and focus on that. So what happens is the urinary bladder is all aligned with the transitional epithelium, and it tends to dip down and include portions of the urethra. So it doesn't just simply stop at the opening into the urethra. It extends somewhat down into the urethra. So that's why we see that listed. So the urethra, the urinary bladder, and extends down a little bit into the urethra. And it's true also with the ureters, which are coming in this direction here. So it does tend to extend up in that direction a little bit as well. So it's just like an overflow, if you will, from the urinary bladder. Function allows stretching and also provides protection. So one of the things that you may not realize is that when you produce urine, it could be very acidic at times, depending on what you're eating. And as a result, where you're storing that material can actually come under a lot of stress from a very acidic environment. So a layer of mucus sitting on top of these cells will protect them from that acidic environment. Let's go ahead and take a look at that. And here we go. That's not right. I'm starting to get confused. I must be hungry. Here we are. And in English. Nicely done, Martin. Okay, so there it is. Transitional epithelium, and this is the unstretched condition. And remember, I talked about how these cells are going to be larger than down here. We covered that in the lab, and we can see that play out here. So transitional epithelium, and then we extend down a little bit in this direction, okay, and a little bit up in this direction. Okay. So this will be pseudo-stratified epithelium. Pseudo-stratified columnar epithelium. And it comes in two forms. Again, non-ciliated and ciliated. Okay. Location for non-ciliated would be the epididymis, which happens to be one of my favorite terms in anatomy, epididymis. It sounds so great on the tongue, epididymis. And that's a structure that's part of the male reproductive system. Also, the large ducts of many glands. And parts of the male urethra. Okay. The ciliated form are airways of most of the upper respiratory tract. Function, absorption, and secretion. Absorption and secretion. Let's take a look at a couple of examples, and then I'm going to go eat dinner. All right. So absorption and secretion. And here our first example, and I think perhaps our only example actually, is the ciliated form. And there we go. So here we go. Pseudo-stratified. So if you're looking at this, doesn't that appear to be stratified? You've got lots of nuclei all stacked on top of each other. So you're saying to yourself, oh, that's got to be stratified. In fact, it actually isn't, because we get some shorter cells and longer cells, and that produces the stratification appearance. But it is a single layer, in fact. We do have the cilia, as you can see. Now, one thing to keep in mind, if you're thinking about cilia, then you have to be thinking about moving something. That's the whole purpose of cilia. So when you're thinking about where these ciliated tissues are located, think about places where you have to move some sort of substance. In this case, it's mucus, because it's in the respiratory tract. So we're moving mucus across that surface, and that mucus has a number of functions in the respiratory tract. You'll learn about that when you do the respiratory system, which is in 40B. So we have this constant beating of cilia that allows the movement of that mucus up and out of the respiratory system and up towards your mouth. Okay. So that is the story on pseudostratified. All right, ladies and gentlemen, I'm going to call it a day. Thank you for coming for session 2, and I will see you for session 3 as soon as you look at it.